So welcome back to the second edition of Corpus Curiosum. This is the second talk of this series, so you probably already know us, but let me introduce a bit the project for those who are new here. First of all, we wanted to thank a lot our sponsors, the British Neuroscience Association and the FENS, because without them, the second edition wouldn't be possible at all. Some important notes is that, first of all, we are going to record this introduction and the main talk because we might publish it in the future. But don't worry, because we are not going to record the discussion. Regarding the questions and answers, so please feel free to ask whatever you have in mind. This is a very safe space, so we want you to share with us whatever you have in mind. Everything is important, and there are no silly questions. So you can use the chat to ask questions during the whole talk, and then Mariela will answer them at the end of the talk. The length of this talk is 1 hour and a half, so we will close this session at 4:30 p.m. UK time, but of course you are free to leave whenever you want. As you can imagine, we were inspired by this structure here, the Corpus Callosum, to pick the name of this project. And this is because in the same way that the Corpus Callosum is connecting both hemispheres of the brain, we are aimed at connecting many young neuroscientists all over the world. A bit about us. As you know, we are 3 neuroscientists in different stages of our careers, and we are also based in different cities. Ines is in Madrid, Faisal is in London, and I'm in Zurich right now. And about you, only for this talk, you signed up from 32 different countries and more than 100 institutions and companies. So big thank you also for you. And finally, the most important part of this introduction is the biography of Dr. Mariela Trinchero. She is a biochemist by the University of Buenos Aires, and she did an internship in the Department of Human Physiopathology, where she studied the modulation of the noradrenergic nervous system by neurotrophins in the hypothalamus. Then she was also working at the Institute of Pharmacological Studies, also in Buenos Aires, digging into the role of the environmental enrichment in depression. And finally, she joined Alejandro Schindler's lab and Dr. Arbus' lab in France to develop her doctoral thesis. And currently, she is doing her research as a postdoctoral fellow in Schindler's lab as well, and studying the neurogenesis in the aging brain. Today's topic is new neurons in the adult brain, breaking the dogma. And well, we invited her because probably most of you have heard that neurons cannot divide at all in the brain, and this is of course a dogma. So we wanted to know what Mariela thinks about that.
So hello everyone, thank you for joining. I wanted to thank the Corpus Curiosum team for inviting me to give this talk. And today, as Salva told you, I will talk to you about new neurons in the adult brain, breaking the dogma. So I am sure that even though you answer pretty well the poll about this, you at some point in your life have heard that we are born with a certain amount of neurons and then we die with the same amount of neurons. And why is that? Well, here I will introduce you to Ramon y Cajal. He was a great scientist. He was a Nobel Prize winner, actually, and he's considered the father of modern neuroscience. So, in the central dogma of neurobiology, he established that in the adult centers, the nerve paths are something fixed, ended, and immutable, that everything may die and nothing may be regenerated. But he also had the humility to acknowledge that it is for the science of the future to change, if possible, this harsh decree. So for a long time, the belief that there was the possibility to generate new neurons in the adult brain was not an option. But then in the 60s, Joseph Feldman injected rats with radioactive thymidine, which will incorporate into the cells upon division, and found that both in rats and cats, he could see cells proliferating in the brain. And then Michael Kaplan in the 70s combined this radioactive thymidine tool with electron microscopy. And this was really important because he confirmed that the cells that were being produced in the adult brain were actually neurons. But still in the field, scientists remain a bit skeptical to this idea because it was really controversial. Then in the 80s, Fernando Nottebohm, who studied songbirds, also using these techniques, found that adult canaries also had proliferating cells in the adult brain. But because canaries are not mammals, the field remained still skeptical. But then in the 90s, Elizabeth Kuhl injected monkeys with BRDU, bromodioxyuridine, which is a thymidine analog that will also incorporate into the DNA upon cell division. And by doing colocalization with NUN, which is a neuronal marker, she confirmed by using confocal microscopy that there were new neurons produced in the adult monkey. So at this point, you might think, okay, enough with the animals, right? What happens with the human? Well, I will show you two really nice papers. The first one is from Erickson and colleagues, in which they studied samples from cancer patients which have been administered with BRDU in order to assess the progression of the tumor. So by analyzing postmortem tissue, they found that there were cells proliferating in the adult hippocampus. And then last year, there was this beautiful paper coming out from the group of Maria Llorens Martin in Madrid, where they studied double cortin positive cells, which are indicative of immature neurons. So as you see here in red, all these neurons were born in the adulthood. They also show that in Alzheimer's patients, the number of DCX positive cells decreased with the progression of the disease. So as you can see, adult neurogenesis is a pretty conserved process that goes along all these species I'm showing here, including the human. But in order to study in detail adult neurogenesis, we take advantage of mouse models. And in the mouse, it has been described that there are two restricted zones where adult neurogenesis takes place. One is the subventricular zone here in purple, which gives rise to interneurons that will integrate into the olfactory bulb. And the other one is the subgranular zone in the hippocampus, which will give rise to dentate granule cells. So, because today I will focus on hippocampal neurogenesis, I will tell you a little bit about these structures because I have mentioned it before, and I want to tell you what is the function of the hippocampus. So the hippocampus has been reported to be involved in learning, memory, spatial navigation, and the emotional control of behavior. And as you can imagine, having these abilities requires a lot of plasticity, right? So here I show you a scheme of the hippocampus organization. You can see here that is a banana-shaped structure here in the adult mouse brain. And if you take a coronal section here in this scheme, I can show you the three synaptic hippocampal pathway. So this is a very simplistic view of the circuit, which is basically formed of three layers of glutamatergic neurons, which are dentate gyrus, CA3, and CA1. So basically what happens is that sensory information comes from the entorhinal cortex, impacts into dentate granule cells, and then they send the information to CA3, and CA3 pyramidal neurons send the information to CA1, which will send it back to the entorhinal cortex to close the loop. So, so far you might think that this circuit is really like simple, and actually it's not. It's really complex, and why is that? The thing is that in this scheme, I am not showing you that the hippocampus has a lot of GABAergic interneurons that will set the tone of the circuit because they will impose inhibitory inputs that will modulate circuital activity. They also have, there is also glutamatergic interneurons, and also there are inputs coming from other regions of the brain, even long-range regions. And what else? Well, we came here today to talk about adult neurogenesis. So new neurons are being formed here in the dentate gyrus. So all the time throughout life. So as you can imagine, the addition of units that will process the information will for sure change the way the hippocampus will work. So what are these neurons good for? Well, there are several papers addressing this question, but I think it's not closed yet. But in experiments where there was an ablation of adult neurogenesis, animals were impaired in spatial memory tasks. Also, adult neurogenesis has been involved in a process called pattern separation. And what is this? So, pattern separation is the ability to take two very similar inputs and decide that they are really different outputs. So it is the ability to discriminate two very similar stimulus. And that is what is thought that adult neurogenesis is doing. So in order to study adult neurogenesis, we have a few tools that I will going to present to you now. One is this genetically modified mouse line. And basically what happens is that you inject tamoxifen and all the neural progenitor cells that are expressing ASCL1 will start to express tomato, which you can see in red for sure. So in this way, we are allowed to label cells in red and study them, right? So basically, I show you here, let me tell you a little bit about this image. In Neurons, you have a neuronal marker, and you can see here the three layers I talked to you about before, the dentate granule, the dentate gyrus, the CA3 region, and the CA1. So basically here is the dentate gyrus, and here in this particular zone is the subgranular zone where neural progenitor cells reside and give rise to new neurons. So these new neurons will integrate into the granule cell layer and will become fully mature and start connected with the entorhinal cortex. So here I show you a beautiful neuron, which is already mature, as you can see. And this neuron has dendrites that are here, and they are like input cables. And here they receive the input coming from the entorhinal cortex in the molecular layer. So how is this connectivity work? So basically, the entorhinal cortex axons impact into these tiny specialized structures, which are called dendritic spines. So this connectivity is glutamatergic. And basically, these new neurons project their axons, which are the output cables of the neurons, through the hilus towards CA3. And then CA3 will give the information to CA1, and then CA1 back to the entorhinal cortex, as I told you before. So another way to study adult neurogenesis is the stereotactical injection of a retrovirus expressing the green fluorescent protein. And this retrovirus will only infect neurons, cells that are in division at the moment of the injection. So basically, this allows us to label new neurons with a green marker or whatever construction you want to put into it, and then we can do electrophysiological recordings or confocal microscopy, as we also can do in the ASCL1 tomato mice. Here I show you how we take advantage of this tool to study how neurons start maturing and their dendrites start going through the granule cell layer in order to reach the molecular layer where they will find their axons the axons of the entorhinal cortex to which they have to connect. So, as you can see, a 4-week-old 21 dpi neuron looks really morphologically mature. So these tools have allowed us and other labs in the world to really describe in detail how is the development of adult neurogenesis. So in the first stages of maturation, these neurons express double cortin, as I told you before, and they migrate radially into the granule cell, and they start receiving GABAergic dendritic inputs, which at this point will be excitatory, while GABA is well known to be an inhibitory neurotransmitter. Then, as time goes by, GABA will become inhibitory, and the cell will start to receive glutamatergic inputs from the entorhinal cortex. Then, when these neurons are 3 to 4 weeks old, something really interesting will happen, which is that they will start receiving a high degree of glutamatergic inputs, but the GABAergic inputs will be a bit restrained at this point. And what happens with this? The result is a high excitatory inhibitory balance. And this makes these neurons to be highly plastic and really excitable. But then by 8 weeks, they go through the final step of neuronal maturation, which is to receive somatic GABAergic inputs, which will decrease their excitability, and then they are fully mature and integrated into the circuit. But this is not like a static process. This is really dynamic, and actually you can modulate the velocity of the maturation. And how can you do that? Well, for instance, I can show you here a 21 dpi neuron looking like this. It has a dendritic tree length of about 400 microns. But then, if mice start running and run for 3 weeks in a running wheel, you can see that the dendritic tree length has increased at 50%. Other way that has been described by people in our lab to modulate neuronal development is, again, we have here the 21 dpi neuron, and what Diego Alvarez and Damiana Giacomini did in the lab was to expose animals to an enriched environment, which I will show you later, from the day 9 to the day 11 of neuronal development. And that elicited an acceleration in neuronal maturation and integration. So as you can see, adult neurogenesis is a highly plastic process. But not only can you accelerate development, but you can increase or decrease the number of newborn neurons depending on some factors. So there are physiological factors such as physical exercise, enriched environment, and learning, which can increase the numbers of new neurons generated in the adult brain. But there are also pathological factors like epilepsy, but in this case, adult neurogenesis is a bit aberrant and is not like completely normal. But then there are another factors that will decrease the numbers of neurons, such as aging, depression, stress, or Alzheimer's disease. So today, in the second part of my talk, I will focus on aging. And here I want to address the fact that this might represent another dogma, claiming that perhaps during aging, there is no plasticity, right? Because it's something that is like an established idea that once you get old, there is no plasticity in the adult brain. So in the lab, we wanted to address these two questions. What is the quality of the neurons generated in the aging hippocampus? And also, do these neurons conserve any kind of plasticity? So to do that, we injected a retrovirus expressing GFP, as I told you before, in 2-month-old mice, 5-month-old mice, or 8-month-old mice, which are middle-aged. So then you can sacrifice mice at different time points to study how is their morphology or morphological parameters. So basically, if you focus on 21-day neurons, you can see that in young animals, neurons look pretty mature, but as the animal gets old, these neurons have a less arborized dendritic trees, and then in middle-aged animals, 8-month-old animals, you can already see some features that correspond to really mature neurons, such as these processes that are parallel to the granule cell layer. And you can also see that this dendrite is not even reaching the molecular layer, right? So you could assume that this neuron is not connected to the entorhinal cortex. So in accordance to that finding, we found that actually these neurons have no dendritic spines at all, which makes sense because it looks like they are not really connected into the circuit. If we wait longer, we can see that these neurons actually do end up maturing and integrating into the circuit. It's just that it takes a bit longer. So basically, we concluded that aging delays morphological maturation, especially during the first weeks of development. So we wonder, why could this be happening? So we know that local activity is really important for the hippocampus, and all the cells in this circuit are sensing all the time the electrical activity of the circuit. So basically, to address this question, we studied ARC expression in the granule cell layer. ARC is an immediate gene that starts to express when neurons are active. So whenever you see here a red neuron, you can assume that that neuron was active at least a little bit before the animal was sacrificed. And as you can see in this quantification, the density of R positive cells decreases with age, meaning that the local activity is decreased with age. Then we study BDNF levels. So the brain-derived neurotrophic factor is like a fertilizer for neurons, right? It improves, it promotes survival, dendritogenesis, synaptogenesis, and it is really the most abundant neurotrophic factor in the brain. So we try to quantify these BDNF protein levels by doing Western blot, and we also saw that BDNF levels decrease with age. So far we have a delay in the development of adult neurogenesis that is supported by a low activity circuit and also a niche with low BDNF levels. So next we say, okay, so if these are the factors supporting this slow maturation, we might be able to modulate them. So we know that physical exercise and enriched environment are hippocampal activity modulators. So first, we try to address the question of what happens with physical exercise and are these neurons able to modulate with this stimulus. So to study this, we injected again 8-month-old mice with the retrovirus first GFP and waited 21 days. And one set of animals were put to run in a running wheel. And I must tell you that mice run a lot, like 10 kilometers a night. So yeah, good for them. And then we sacrificed mice at 21 days post-injections. So as I showed you before, in sedentary mice, you can see that neurons are not really mature. They don't have mature features in terms of their dendritic tree. But then, and this was really surprising, when we looked for running mice, we could see that the dendritic tree length was amazingly increased in a really significant way. So I am sure after this you will all go to run tomorrow. And here I show you a mature neuron born in a young mouse, and you can see the similarity between these two neurons. So here I have the quantification showing that the increase comes from 100 micrometers dendritic tree length to 500. So this is a really highly significant difference. So what happens with the dendritic spines? Well, in the same experiment, we took confocal images of the dendritic spines, and as I told you before, 21 DPI neurons from sedentary mice are not connected at all. They don't have any spines, but running animals had their dendritic shaft covered in a lot of spines that actually was more than the number that we found in 21-day-old neurons in young mouse. So as you can see, the level of plasticity of these neurons is really high. As you can see here in this graph, showing that it's an all or nothing situation, right? The control neurons have no spines whatsoever, and the running group has a lot of spines. So basically, I show you that running accelerates neuronal maturation in the Asian brain. So what about enriched environment? So here I show you a video of how enriched environment looks in our lab. This is a larger cage with a lot of like toys and tunnels where they like to explore and go through, and they even have like a cute house to live in. And so basically we ask the question of whether enriched environment can modulate neuronal development for 3 weeks, or if they were able to respond to small periods of enriched environment of 1 week. So what we saw is that in a similar way to running, exposing mice to 3 weeks of enriched environment actually increase the dendritic tree length and the spine density in a significant way. But what about short periods of enriched environment? So we always study 21 DPI neurons and found that there is this critical window from day 7 to day 14 of neuronal development in which these neurons are able to respond and extend their dendrites and integrate faster into the circuit. So I am talking to you about integration, but I am only showing you morphological features. So what about functional features showing that they are actually integrated into the circuit? So for that, we need to do a little experiments that are a bit harder, but they are really cool. So these are electrophysiological recordings of new neurons. And basically, we sacrifice mice and take their brains to put here in the vibratome, and we make acute slices like this one, which we keep alive for the whole day to record neuronal functional properties. So these are really cool experiments that I really love doing. So this is the setup where we perform these experiments. And this is how we look at the slices in our setup. And this is the pipette we use to patch these cells in order to study them. So basically, we wanted to know whether neurons exposed to enriched environment from the day 7 to the day 14 of neuronal development were able to integrate faster into the circuit by this electrophysiological approach. So we addressed the spiking, which is the ability of these neurons to generate action potentials. So in the control, you cannot see any spike, which makes sense because these neurons look like this, right? So maybe you wouldn't expect this neuron to be able to fire action potential. But then when we look at 21 DPI neurons that were exposed to enriched environments for 1 week, we could see that these neurons were able to fire several action potentials, which also makes sense because if you look at the morphology, they look really more mature than the control neurons. Then we assess the connectivity. So the connectivity is telling us that the frequency of events where neurotransmitter binds to the receptor of the neuron that we are recording. So here I show you this is current, this is the baseline, and basically there is no deflection of the signal. So this neuron is not connected, which makes sense again because of the lack of spine, dendritic spines, which might tell you already that these neurons are not receiving glutamatergic inputs. But then when we study neurons from mice exposed to 1 week of enriched environment, we found a lot of deflections of the baseline, meaning we quantified this frequency of excitatory postsynaptic currents, and we found a significant increase from basically 0 to 1.5 hertz. And this also makes sense because these neurons do have dendritic spines, so you would expect them to be to have some degree of connection. So basically, I have told you that 7 days of enriched environment exposure accelerates integration of cells in the Asian hippocampus. So as you can see, the level of plasticity that we found in the Asian hippocampus was, I think, even larger than we have expected. To conclude, the main conclusions of this talk are that the generation of new neurons is a complex, dynamic, and extraordinary form of plasticity taking place in the adult brain. Also, in the Asian hippocampus, where major changes are not expected, basal activity is low, and developing neurons appear really mature, brief periods of physiological stimuli trigger high levels of plasticity that even exceed those seen in young animals. For instance, if you remember what I showed you in young running animals, you can see that the difference here is 50%, while here is 400%. So the ability of these neurons to respond and integrate into the circuit is really amazing. So neurons born in the aged animal are a source of great deal of plasticity, and the circuit is also plastic enough to integrate these neurons. So what is the take-home message of this talk? So I think basically we broke 2 dogmas today. So we thought that there might be no neurogenesis, but now we know that there are a lot of neurons being generated in the adult dentate gyrus. But also, we thought that maybe there was not much neuronal plasticity in the Asian brain, and now we know that the Asian brain has high levels of plasticity. So I think a nice message of this talk in general is that we as young neuroscientists, we need to be flexible, try to think outside the box, don't hold on to one idea, and be critical with our own work, and stay curious always to keep breaking dogmas. So I want to thank all of you for your attention. I want to thank our amazing group leader, Alejandro Schindler. You can follow us on Instagram if you want. And this is the wonderful group I am lucky enough to work with. And I want to especially thank Maggie Herrero, who was my partner through all this Asian adventure. So now I am happy to take questions.